Welcome to today's video. In this one, we are going to be just looking at the structure of one muscle fiber and more specifically looking down into what makes up a sarcomere, which are the functional units of a muscle fiber. So first things first, muscle fiber is just a name for a muscle cell. So one muscle fiber is one muscle cell. And these types of cells are very unique. If you remember from when we were talking about tissues, skeletal muscle tissue, each cell has a lot of nuclei. And the reason for that is that during development and as you work out more and your muscle fibers build and are repaired, they fuse together with new stem cells. So that's the reason why each of these cells have more than one nuclei, and really they have a lot of nuclei. So the name for the membrane that surrounds a muscle fiber is the sarcolemma. So you're going to see this prefix quite a bit, okay? So sarcolemma is just the special name for the cell membrane of a muscle fiber. The sarcoplasm, again, this is the cytoplasm, but specifically for a muscle fiber. And the sarcoplasm is unique from other cells because, again, there are many nuclei, plus there are a lot of mitochondria to help to drive the production of ATP through cellular respiration. So within the muscle fiber, we have all of these little bundles, okay? They're bundles of fibers, protein fibers. And the main protein fibers that make these myofibrils up are actin and myosin. So we're going to be talking a lot more about each of these here in a little bit, but actin and myosin, keep those in your mind. Those are the main components of our myofibrils. Okay, and then if we look closely at a myofibril, we have these little functional units, right? They repeat all the way down the length of the muscle fiber. And these functional units are called sarcomeres. And they produce these striations that we have talked about previously as well in the tissue unit. Okay, so another special structure here in the muscle fiber is the sarcoplasmic reticulum. So this is like the endoplasmic reticulum, but again, specialized for our muscle fiber. So these are membranous channels, just like endoplasmic reticulum. And these channels surround each of the myofibrils, as you can see right here. Okay, so it's this orange structure that almost kind of looks like a web just surrounding the myofibril, and the sarcoplasmic reticulum surrounds all of these myofibrils. Okay, so if we take a look here at the sarcoplasmic reticulum, we can see again repeating units down here. These are called triads. So if you look, we've got 1, 2, 3 components of the triads. So right here in the middle is called the transverse tubule. And this is actually continuous with the sarcolemma. So the sarcolemma, again, is the cell membrane. And the sarcolemma actually pinches down into the muscle fiber and forms these transverse tubules surrounding each of the myofibrils. So the reason why this is important is because these transverse tubules contain extracellular fluid. So what would normally be on the outside of the cell is actually able to come down into these transverse tubules and flow close to the sarcoplasmic reticulum. So the activation of muscle fiber is it goes through this transverse tubule and then will activate down here in the other part of the triad. Okay, so these other two parts of the triad are called the cisternae, and they are just larger portions of the sarcoplasmic reticulum. As you can see, it's kind of thin here, but it gets a little bit thicker as you get towards the triad. So the main thing about these cisternae is that they contain large concentrations of calcium ions. So when the cell is activated, then the sarcoplasmic reticulum here in the cisternae are going to release all those calcium ions. Okay, so now let's talk about a bit about the sarcomere. So they are repeating patterns. They are the functional units of our muscle fiber. So when the muscle fiber contracts, it's really happening at the sarcomere level, okay, end to end, all the way down the muscle fiber. So each sarcomere is made up of thin filaments and thick filaments. The thin filaments are actin, and the thick filaments are myosin. The thin filaments contain a few other proteins too, but they're primarily actin. The thick filament also is slightly composed of another protein as well, but again is primarily myosin. So these sarcomeres, the overlaying of the actin and the myosin produce these stripes that we saw whenever we were looking at the muscle fibers under the microscope. Okay, so in reality, a sarcomere is three-dimensional, and they fit together in this bundle. Okay, so all of these sarcomeres side by side, end to end, they form this bundle all the way down in muscle fiber. So it is three-dimensional, but we are going to be looking mainly at a two-dimensional depiction of our sarcomere. Okay. So we take a look and we classify a few of the parts here of the sarcomere. The end of the sarcomere is the Z disc. Okay. So we've got the Z disc here. We've got the Z disc here. It is bound to the actin. It's also bound to this protein here called titin, which holds on to the myosin. Okay. So again, the Z disc are just the boundaries at the end of each sarcomere. The I band, okay, the I band is right here, where we only have this thin filament or actin. The A band, okay, is from one end of the thick filament all the way to the other end of the thick filament. The H zone is right here, in the middle of our sarcomere, where we only have thick filament. And then the M line is the very center of the sarcomere here, where the M line is just the very center. It helps to hold the myosin in place. And yeah. Okay. So let's take a look at each of these filaments, starting with the thick filament. The thick filament's a little bit simpler. It is mainly composed of the myosin. So if we're taking a look here, all this purple, this is all myosin. So these are strands of these protein filaments all bundled together. Each molecule of myosin is made up of two protein strands twisted together. Okay, and then at the very end, they have these globular heads. Okay, and these heads are going to be what attach to the actin and form the cross bridge. So at the other end of, at each end of the myosin or the thick filament, we have that little kind of, looks like a spring protein called titin, and it helps to hold the myosin to the Z disc, and it also compresses whenever the Z discs are moving closer together during contraction. Okay, so this right here, the yellow, that's the titin. Okay, now the thin filament. And so we have here, 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 and here. Okay, so the thin filament is towards the margin of our sarcomere. Okay, at least comes from the margin, and during contraction, it's going to move towards the center, towards the M line. Okay, so actin is made up of globular molecules. Okay, they're ball-shaped, essentially. They are attached to one another. They form a double helix-shaped formation, kind of like DNA. Now, here in our actin, what we can't see, because these strands of a different protein are covering them up, but each actin molecule has a binding site for the myosin head. Okay, so during activation, this myosin head is going to be able to attach to one of those active sites when they are unlocked. Okay, so during rest, okay, or not during activation. The actin active sites are being covered up by a troponin tropomyosin complex. Okay, so this is a combination of two proteins. So the tropomyosin is what is really covering up the actin active sites. Okay, so it's these rod-shaped proteins that are covering it up. And attached to the tropomyosin is another protein called troponin. And troponin is made up of 3 protein subunits, as you can see here, and it helps to hold the tropomyosin in place. So the troponin can get unlocked. So you can think of you can almost think of the tropomyosin as like a door, right, that blocks the way. The troponin is like a lock. And then what we will see later is that calcium is going to be the key that unlocks the troponin, okay? So we've got our door, we've got our lock, and during rest, okay, when our muscle fiber is not activated, then these are going to be locked in place. But during the activation of our muscle fiber, when calcium comes in, calcium is going to unlock this and shift the tropomyosin off of it so that the myosin heads can then attach and then go through a whole series of events, okay? But that is going to come later this week. So that is all for this video today. I hope that you were able to learn something from this. Make sure that you have taken notes because we're going to use this in class tomorrow. Have a good day.